can I do something to actually slow this disease down?
9 years ago, before Eric entered that clinical trial, Dr. Glass told us where this was headed. Dr. Glass is working to turn off the genes that cause the disease.
We're going to start a trial probably later this year where these things will be put into the spinal fluid of patients, and hopefully they will target that abnormal gene and turn it off. Boom, it's like a switch that can turn it off. We're going to get a cure.
The question is, how do we take a genetic treatment that works for 2% and make it work for everyone else? At Emory, physician scientists and researchers are working to find the signals that lead to treatments.
I'm looking for biomarkers. Mostly I'm looking for ALS biomarkers that we can find in blood.
So this is a combination of the...
I try to understand, as I mentioned, trying to understand the disease mechanism of the underlying ALS. ALS is like 10% you have a family history. So meaning there's some sort of genetic mutations. And there's a 90% of ALS patients that are sporadic, no family history.
My research is focused on early diagnostics for the detection of neurodegenerative diseases, a broad range of neurodegenerative diseases, including Alzheimer's disease and ALS.
Right. What do you do with it?
For decades, trials asked one question: does this drug work? Now Glass and his team are asking another: what can we learn when it doesn't?
We need to figure out, because there may be drugs in there that could have worked, but we didn't give them enough, or it didn't get to where it needed to be, or it's not in the right areas of the nervous system. And I have a nice slide that I show of dozens and dozens of ALS treatments that have failed over the years. And I would argue we don't know why.
He had a paper on multiple...
One of the researchers studying why these trials failed is Zach McEachin, an assistant professor of human genetics.
Why did the trial fail? Did it fail because it's the wrong target? Did it fail because the drug or the molecule, this ASO, did it not go where it needed to go?
That kind of analysis can reveal the right patients, the right timing, and the right targets for future therapies.
Galectin-3 is the biggest immune response.
McEachin has also developed an antisense oligonucleotide to potentially treat ALS.
I'm excited by the data. We just got new data in today that verified our first findings.
Because this gene that we're attacking.
But first, a quick lesson.
This protein called TDP-43, okay? It's in all of our cells, everybody has it, it's in the nucleus, and it's important.
TDP-43 is a normal protein we all have, but in 97% of ALS patients, it ends up in the wrong place. TDP-43 normally sits in the nucleus, the cell's control center, editing genetic instructions. In ALS, TDP-43 leaves the nucleus and forms clumps. Without that editor, the cell reads hidden junk segments called cryptic exons and begins building proteins incorrectly or not at all. Antisense oligonucleotides are tiny synthetic genetic molecules designed to block those junk segments and restore the correct instructions.
Using this ASO not only corrected the one protein that we were targeting, that is downstream of TDP, but it also corrected other proteins in the, in the system, like in the cell.
We have a drug potentially, or a molecule that Zach has invented that actually can take 65% of these proteins, and there are hundreds of them, and shift them back here. Not one at a time, but a whole bunch of them at the same time. We've tried to prove it doesn't work, and we can't prove it doesn't work. And so we're moving forward.
We were quite excited because we said, if we can do this in a model system, could we give patients with ALS this drug, and could that restore the function, and hopefully they would get better.
The goal? Give these therapies early when warning signs first appear. Glass hopes to begin human trials within 2 years.
The idea would be, with one potential intervention with an antisense oligonucleotide, we could potentially correct a majority of the protein abnormalities that are being caused by this loss of TDP-43. This is the closest I've ever been to being so close to actually taking something from the laboratory and giving it to a patient. I've never been this excited. This one is a game changer, because it's going right at what I think is the basic core of this disease.
Knock, knock. All right,
Back at the Emory ALS Clinic, this is why the work continues.
Doing science is not easy because we have a lot of failures. And when I feel that I'm down, I always talk to Dr. Glass. I will go to his clinic and just to see the patients and really give me a lot of strength to really continue my work.
Every story is different. Some are about care, some are about connection, some are about purpose, and some are beginning to be about treatment. This is what progress looks like now, not one miracle, but new paths forward.